Thank you very much, and welcome to my talk. So I'll be talking about how to visualize and modify DNA origami structures in different scales and dimensions, and I will describe a way to, I will describe a map that allows the interaction in this abstraction map. So first of all, what is DNA origami? The main idea of DNA origami is to facilitate DNA as a building material to construct nanoscale objects. And the way this is usually done is we extract a long DNA single strand from a bacteriophage that you can see here. We call this single strand scaffold. And this scaffold is pulled together by so-called staple strands, very short single strands. And they bind to different domains on this scaffold strands. In this way, the scaffold strand can be folded. And we do this by programming the sequences of these staple strands. Now, if you program a lot of these staple strands and they bind to very specific regions on the scaffold, you can basically program the self-assembly of this scaffold strand. And it can self-assemble itself to any arbitrary structures, in this case into a rectangular, as you can see. This is the in silico design process, and after that we can export the sequences and we send them to the lab. And in the lab, so inside these tubes, like the DNA has been synthesized. And in the last step, our biochemist here in the lab, she does the self-assembly of these structures. The problem is, this is very expensive. So we really need to make sure that the sequences that we send to the lab are correct. So this is to quickly show to you the foundation of DNA origami. So here on the left side, you can see the chemical structure of DNA. It has a backbone that connects the nucleotides into a chain, and this is called the DNA single strand. And inside the side chain, you find 4 bases that are complementary to each other, and this way the 2 single strands form the characteristic double helical structure of DNA. What is very important for DNA origami is the crossovers. That means a DNA single strand can cross over from one double helix into another one, and this way bind 2 double helices together. And if you repeat this, you can bind a lot of double helices together, and this is why this works, and this is how it works. And if you look at the DNA double helix from the bottom, imagine it as a cylinder. The cross-section is a circle, and it will arrange itself in a certain pattern, in a certain lattice. So we have a honeycomb pattern and a square pattern, depending on where the crossover locations are. So as you can see, I needed to use very different representations just to explain to you the very basics of DNA origami. And this is one of the main problems that we have. But first of all, the DNA origami method was very recently discovered by Paul Rothermond in 2006, and there he used very, let's say, manual methods to generate these structures. So, so actually, these are the nanoscale objects using transmission electron microscopy. And so it was just a question of time when the first tool would appear. This was done in 2009 by Sean Douglas. And you can see here a DNA nanotube that we have designed. And it consists basically of multiple views where you have a 2D view using DNA schematics where you can design these objects. So, but this is pretty much the state of the art. It is very, very wide employed. So in our project, in collaboration with the Austrian Institute of Technology, our goal is to use this technology and build a DNA nanorobot. So this is a pretty large structure. What its purpose is to find or to target pathogens and destroy these pathogens, such as antibiotic-resistant bacteria. As you can imagine, this is a very, very ambitious goal, and you can only see a conceptual drawing here. But we have already created some components of this structure. For example, this 3D nanotube. So with very complex objects, maybe you can take it apart into single components that are a little bit simpler. And this is exactly what our domain scientists are doing. But the problem is the state-of-the-art methods, they have never been created for such high-order structures as you can see here. So our domain scientists, our partners, they have to come up with very, very different workarounds. And the problem with the workarounds is that they are to some degree prone to errors. And this is the main problem currently that inhibits kind of the progress in our project. So let me show you quickly how this DNA nanotube was designed. We have very, very traditional DNA schematics that you can see here. And the advantage is that using these 2D views, we can, it's very, very well suited for easy interaction. We get a good overview. But the problem is, since we designed something that is 3-dimensional on a 2-dimensional layout, we have a lot of visual complexity, a lot of visual clutter. Another problem is that we really need to know how DNA works, how DNA nanotechnology works, and these are the constraints that are imposed on these designs. So this, to some degree, makes the design process also a little bit difficult. And in 2D, you have not so much, so you don't have a lot of spatiality. That means what is a neighbor in 3D doesn't mean that it is a neighbor in 2D. But this is why we have 3D views, right? So this is the DNA nanotube. These cylinders represent the double helices. So in 3D, you have very realistic spatial arrangement of the DNA strands, but the problem with 3D is visual occlusion. And also, it is very hard to examine individual strands. But in 1D, this is basically the sequence, the primary structure of the DNA strands. You have very, very clean arrangement of DNA strands. And the advantage here is what our domain scientists are doing very often. They sort these strands. But there's no spatiality. So as you can see, we have advantages and disadvantages in every dimension. And what our domain scientists would like to do, they would like to work in every dimension they like, but currently this is not possible. They can only work in 2D, design in 2D, and maybe get some feedback from 3D. And when they export the sequences to give to the lab, they can also see it in this 1D. So previously we published this work so you can see here the 10 different scales, this visual abstraction axis of scale, where we depict the different structural properties of this DNA nanotube. And we extend this idea with an orthogonal visual abstraction axis, namely the dimension. And so the idea is that we really distribute the complexity of this structure on many different representations, and each representation highlight different aspects of these structures. And the 2D, the 1D, they are obviously only crop outs. So the advantage is we have an integrated view. So we can select any representation we would like. But as you can imagine, now we have many representations. So in order to relate these representations to each other, we have to think about a way to how we can externalize the mental relation between these representations in order to ease the cognitive load that we put on the viewer. So so we render these structures using geometric primitives such as spheres and cylinders, and then we linearly interpolate between these shapes. So we interpolate the shape parameters, we interpolate the positions and colors in order to seamlessly transition between these representations. So if you compare it to the state of the art, this is the state of the art, and this is basically in our map, the part that represents the information that can be found in the state of the art. But we have many more details that we depict in our dimension and scale unifying map, the DimSUM. So let me show you how this works in action. So here we have an action panel, and by moving around this icon, we can move around in our abstraction map in the DimSUM panel. As you can see, by moving around, we can change really the representation. It is seamlessly transitioned between these. So right now we are in 1D, but we can easily move into 2D, into 3D, and you can see this nice folding kind of animation. And so it gives our domain scientists really a good way to work in any scale, any representation they would like. So our hope is that we created here some sort of a comprehensive set of representations that they need. And it's for sure not complete, but it at least creates a space where new representations can be added into this abstraction map. So this is another thing that is very important for our domain scientists. So here we can, in 1D, we can single out each single strands, and then we can transition to 3D. As you can see, next I will decrease the scale so you can see actually the atomic details here. So this is the typical brushing and linking operation, but through transitions. So crossover locations are very important. So a strand should not cross over too many times, otherwise the stability is kind of compromised. And you can see here how these crossovers are highlighted. We can, in 1D, we can sort. So this, or by the different properties like strand length or GC content, which is a stability measure. And you can see, for example, now these are the crossover locations transitioned into 3D. So as I said, our domain scientists, they use our system not for only examination, but mainly for interacting with the data. So to actually model and modify the structure that they are dealing with. And I demonstrate you now here 2 tasks where it was really, really necessary for them to work in different dimensions. The first one is the removing of short staple strands, and the second one is the adding of so-called bridging strands. So in 1D, we can sort by strand length, for example, and here one strand was selected. The reason why we do this is because these short staple strands, they are generated by the state-of-the-art method automatically, but they have to be removed because in the end they only generate cost during the synthesis, but they don't add anything to the structural stability. So this is now removed, as you can see, and in 1D we can see there are only 2 short staple strands left. Another thing, so these horizontal lines, they represent the DNA double helices, the vertical lines represent the DNA crossovers. And so the crossovers, being these vertical lines, they can be very easily identified in the 2D view. As you can see here, they are now removed. And in order to add a bridging strand, we have to somehow see the realistic distances between the strand endings. And as you can see now, we zoom in into the concavity in the DNA nanotube. And exactly here, we want to add a DNA bridging strand. And simply by clicking on Connect, we can specify a sequence that should be created in between. And by clicking OK, we've added this bridging strand. And this is just to demonstrate you that everything we do in any representation in our abstraction map, the effect of these modifications will be propagated to every other representation. So the interaction panel is also augmented with different information or overlaid. So we have here we have the DimSUM interaction panel, but augmented with this heat map. And this heat map shows us how many seconds the user has spent at a particular location. So because we have a big space, interaction can be, or the workflow can be optimized by showing them additional information. They have different means of interacting with this interaction panel, such as snapping to the axis or to discrete locations, or discrete scales and dimensions. Or if they're really interested in a particular location in this abstraction space, they can also define viewpoints that are basically the screenshots at this particular location. So let me show you how this works in action. So now we load a session that we've logged before. And the interaction panel is now augmented. So here we first demonstrate the axis snapping at a particular discrete dimension or scale, or we can also snap to a particular location here, and then it snaps to a line that is parallel to the axis. So this, in the end, optimizes a bit the workflow. And if they are really interested here, you can see how viewpoints are defined, and then we can also move or travel between these viewpoints. So we did case studies. We have one, so since we work really in the project with our domain scientists, so we developed this tool for them. So one domain scientist, she has been using this system here from day one, basically almost every day for more than a year. And she's mostly concerned with the structural motif design. And for her, so we sat down together with them, and they just explained to us quickly, and we logged their behavior. And it is really interesting to see that actually she's not only interested in 3D or in 2D alone, but actually also in the region in between. Another case study that I want to show you a little bit more in detail is done by another domain scientist. And for her, it was really important to add to modify certain DNA structures, to connect them to other structures in the middle of these DNA structures. So for this, she needs to analyze the surface strands. So imagine we have this DNA cage, and in the center we have a target, and to this target, certain DNA strands should be prolongated to add to this target. And in 2D, obviously, this is very difficult to do because we lack the spatial clues. But in 3D, we can see the helical twist. We can see where the DNA ends in the inside of this cage that can be potentially selected for prolongation. And what she did actually is she looked at our visualization, and she has done that before in her method. But what she discovered using our method was actually that one of these DNA strands, they make a helical twist towards the top, so towards the outside of the cage. And this actually shows that this is a bad candidate for the connection. So this reduces her to this one candidate. And in the end, it allows her to design, let's say, more reliable structures. So this is just to show you again how the DimSUM interaction panel works, how we can change the representations of this DNA cage, and we can really go down into the atomic details here. And she can use this to seamlessly transition between every location in this abstraction space. So, of course, our method doesn't come without limitations. So we have currently, we didn't include any simulation data for structural predictions, but we have the space for it. So this would give us a 4D in our multidimensional representations. One complaint of our domain scientists is that the camera is not smart. So what they mean is that they are focused on something, but by transitioning now between these locations, they kind of lose the focused region. So the camera should somehow follow this. But I think that's more of a challenge. So we see it more of a future work. Another thing is the 2D to 1D transition is very cluttered. That means, so we do this linear interpolation of the positions, but a lot of things happening at the same time result that they have difficulties to follow what's going on. So we thought maybe a two-step interpolation where we first straighten the DNA strands and then we do the transition and the rotation. And what we believe is really interesting for the idea of visual abstraction, of abstraction spaces, is the navigation in these abstraction spaces, because we have many representations now. And we want to somehow navigate in this representation. Maybe we can consider it as a topographic map where we can navigate through this space where we have valleys, we have mountains. And our aim is to really, right now this system is deployed on our in-house users' computers, but when we roll this out to other users from other teams, then we hope that somehow this can be used to log behavior, to see where, for example, a user is particularly interested in a specific region, where for a task we might look at specific regions, or so in the end, it gives us ways to somehow tell different users, another user, which representations we are particularly interested in. So in summary, and to conclude this talk, I showed you novel and existing representations for DNA origami structures that are combined in an abstraction space using visual abstraction. So this abstraction space is spanned by two axes, by the dimension and the scale axis, and we can interact with this. I showed you the interaction methods with this abstraction space. And in the end, what is most important for our domain scientists is that they can really apply these abstraction adaptive modifications where they can work in any representation they would like without having to worry how these modifications are propagated into other representations. So thank you very much.
Yes, thank you very much for the nice presentation. We have time for questions, and maybe I start with one. Okay. I was just wondering, so why are you not displaying a preview for what will be the outcome in your interaction panel per default? Because now, for example, here in the video, it's just white without any content. Something like what you were showing on your summary slide.
You mean having the interaction panel already augmented?
Yes, yes.
Yes, because first we need to get somehow the data. Let's say for a particular task, a user is interested in particular representations. So we need somehow to get this information first. And we could do it, for example, in such a way that the user, she's working on something. And then she uses these representations. She already clicked on log. And then she logged her behavior, and now telling another user, well, look here and there. Another user just opens these session files and already knows where to focus her attention to.
But couldn't you pre-fill it with a meaningful default?
I'm not sure, because the tasks that they're doing, they are very, very different. So some users, they are much more interested in the atomic details, so towards the left side, or top left actually. And some users, they really want to work on these in 2D at the high level representations. And it's difficult to have some sort of default presets. But yeah, sure. I mean, if we have like enough data, then maybe we can come up with something like that. But that's a good point, actually, yeah.
All right. Further questions? Yes?
So kind of related on the 2D view. Is there maybe missed, but is there a reason why you just decide not to have sort of like a legend explaining the different axes? I mean, maybe like a graphical simplified form of the different scales.
Yeah, I mean, sure. I mean that would actually help new users to somehow learn this interaction panel that you can see here. The thing is, we are to some degree also like we have our in-house users, and they know how to interact with our software. So in this case, but I think if we give it to new users, definitely we have to add some labels to the axis here.
Yeah, so thanks for a very nice talk. I think it looks very appealing, the system, and apparently this panel is attracting a lot of questions. I also have a question about this panel. So when you were showing the results of this use cases you were working on, you were annotating the panel, and the annotations were mainly in the top right corner, actually.
Yeah.
So I assume that also means they have only worked with those type of representations. Is that correct?
Exactly.
Okay, so then is that not a good selection of use cases? Are there use cases where people would work with all the others, because those two didn't apparently?
So the thing is, right now you can see, for example, other locations highlighted. So I mean, we have our in-house users.
I guess this is you doing a video for the paper, right? I mean,
no, this was actually the result from some use cases. So yes, for a particular task, they need to stay in, let's say, in certain regions in our abstraction space. But that really depends on the task. And here I selected two tasks where it was really necessary to look at the 3D, the structural properties, or to move between representations, dimensions, and so on. But we have obviously like other use cases where we need to work in different regions. But per use case or per task, usually you stay quite local in some areas. But this is like a global map. And depending on what they want to do, they can stay in certain vicinity of representations that they would like to do. Yeah.
Okay, so maybe you could scale into these areas for the use cases, like that you have a different preset, and you only show those parts. That makes it a little bit better explorable, right?
Yeah.
Okay, thanks.
Yes, maybe one last question.
Helvig Hauser, University of Bern. Let me first congratulate you. I mean, this is amazing. The application is mind-blowing, and your work is very, very interesting. I'm interested to understand, how did it come out that your map became such a nice 2D Cartesian space? I mean, I don't see this given to begin with. For me, it could be that for one particular 3D abstraction, you have not only one, but multiple 1D, or maybe none. How does this happen that you have this nice correspondence, that for every point you have something one and just one corresponding point in the other dimensions?
So we first came up with the different scales. Basically, this is the first visual abstraction that we did. And abstraction is a process where we highlight different things along this axis until something becomes very, very abstract. So we designed this first, and we discovered actually these representations, by changing their positional information in 2D or 1D, they exist there as well. But certain things, like for example the helical twist in 2D, they exist on two scales. So these representations would be the same. So in the end, we don't really have 10 scales times 3. The 3D, 2D, 1D, the three things that we have in the dimensions. So it's not really 30, it's like 27 or so, because some things we were not able to actually depict also in 2D. Yeah, that's actually a very good observation, yeah.
And you could have multiples, right? I mean, that often you have multiple choices for abstractions.
Yeah, but for our case, we really sat down with our domain scientists. I mean, I work in this team, and they told me, well, this is what I need. Give me that. And then, we created these representations just for their specific needs that are, I think, these problems, they are prevalent all over the DNA nanotechnology field.
Okay, one final question, and then maybe we'll go for the coffee break, and Haichao will be around for sure to answer further questions.
So to follow up on the question that was asked before, so my name is Anselm Spöri from Rutgers University. How generalizable is this to other structures? The structures seem very orderly, sort of very vertical and horizontal, and that produces these very easily to read and nicely implemented visualizations. So could you take this to any molecular structures?
So right now we work on proteins. So, but I would say for a certain subset of particular proteins where you have a lot of repetitions, we are actually applying this idea there. So protein, you have also primary structure, secondary structure, and all of that. But actually, what we discovered while working on this is these data characteristics that you find in DNA. So we have elongated linear tubular structures. This exists in other data as well. So for example, in cartography, where you have lines representing streets, roads, rivers, and so on. So maybe we can also here transition between the spaces where we have different abstraction levels and so on.
All right. Then let's thank all speakers again.
Thank you.